All right. So as I was prefacing it, I mean, we're going to engage with chapter three, but I think I'll do most of that aspect of things probably a bit more on Thursday when I meet with you. And I'll try to prep together or put together some kind of a PowerPoint with some of this, or at least some slides with this information. But there's a lot of them out there, and I'll send you the one we're going to look at today because I thought it was a nice one. And so we have to start looking at things like these different delays, whether it's in division or interphase or reproductive failure, and all of those things, the curves that we're going to have to look at, the different kinds of damage. So I thought, presenting you as we start to get into sort of the real heavy duty stuff, which will be forthcoming. But at least for now, I did want to go over a little, just what I didn't get to last time I was with you was the cell cycle, because now we're going to see the relevance of that. And as I was thinking about it this morning on the way in, why? But I mean, it's interesting. Therapeutics, and that's my background is clinical, treating people with illnesses, infections, things like that. Thankfully, I did not have to deal a whole lot with cancers other than perhaps skin cancers more than anything else. But I did order a lot of things in your areas of expertise, variety of advanced imaging and scans. So this is really, where we, and to be fair, we had less of it available then than we did now. I mean, when I started in practice, they didn't know about MRIs. Do you train in that also? Because when my first experience with an MRI, which gives you an idea of how old I am. Sorry, you say this stuff. When I was a junior student at Seton Hall University in South Orange, New Jersey, the MRI was a device that we used in chemistry. Okay, to be able to analyze organic chemicals. That's what its basis was, magnetic resonance. It wasn't until the age of computers that they were able to put those together, computer enhanced the spin on protons, which were filled with water and organic compounds, to begin to use nuclear magnetic resonance terminology. So, I mean, it's just, we had a little device that we dissolve some stuff in and create a magnetic field around it, and we didn't have to worry about it, pulling your ring off or, having metallic hardware in your body and being impacted. So it gives you an idea of the changes. And when I started in practice, I started my doctorate in 77. So that every year you began to see a greater impact of these kinds of things. As the technology improved, our understanding, radionuclide compounds, et cetera, et cetera. Okay, so background-wise, it's the advances in technology have really increased our diagnostic and therapeutic ability. So, I utilized it more diagnostically as opposed to be utilizing it more therapeutically. It was not so there weren't the kind of things that I would be ordering, but it's interesting because that's where my focal point of experience is from. So, nevertheless, with that sort of jumping off point, I'll go over here to the cell cycle. I'm sure you had this before. It bears reviewing. This is chapter 3D. Hopefully, I'll be able to see it wherever I put it. And when I teach A&P, and I do it every semester and teach this every year, I do this for the nursing students. This is something I spend a very large amount of time in. And it's to understand this is the critical phase, because when I say it's a two-edged sword, you have not only are you going to be treating cancers, but you can create cancers. That's the idea of the two-edged sword. And it's true with a lot of therapy. You can create damage, you can fix damage, and you can create damage at the same time. So you have to exercise an extraordinary amount of care and due diligence in all of these things that are there. But really, what I understand is it's all about reproductive cycles. So we're looking at effectively, if you wanted to simplify it, to me, cells that reproduce rapidly, read that as cancer cells, they gobble up whatever is in their environment. So because they're doing things so rapidly, they're and creating inordinately large amounts of DNA, breaking it down, building it back up, breaking it down, building it back up, replicating it, undergoing mitosis, is what this cycle is about. They are extraordinarily susceptible. Where cells that do not do this, that have effectively done what we call exiting the cell cycle, are really, relatively speaking, refractory. They'll undergo a certain amount of damage just because of the nature of what radiation does to the surrounding tissues, but it's not destructive in nature. So more about that as we go along. So when we look at the cell cycle, it's just like a big circle. That's all it is. Okay? And it undergoes really two areas. The area that we focused on, and most people focus on excessively, is what's called mitosis, the mitotic phase, which means nuclear division. It's really, at that point, it's really the mechanics of the cell replicating itself. First, so the mitosis, by definition, is the nucleus replicating itself. And cellular division is something called cytokinesis, though we tend to group the terms together, probably erroneously. So in the mitotic phase, what we see is we see the chromosomes appear, we see certain changes that are characteristic of it, which I'll scroll through very, very quickly to show you what they are. But that's not really where the impact of what radiation has. It impacts it indirectly. So what it does to the cell, it's reflected in what goes on in the cell cycle, or not even entering the cell cycle, or entering mitosis rather. Interphase, the big player. As a when I first learned about cells, when they first elementary biology, they drew a picture of, maybe you probably the same thing. They drew a picture of a cell, a rectangle, a circle. Oh, it's a cell. Everybody. And whenever they talked about this, when they started to talk about mitosis, that's when the cell is really busy. And nothing could be further from the truth. Most of the cell's activities are carried on in what you and I call interphase. So you see it here. The period from cell formation, when the cell effectively starts, and it just keeps redoing the cycle. And it's a question what we will look at is the timing that goes on, because different cells do this at different times. Some do it very, very slowly. Some don't do it at all, and some do it very, very rapidly. And certainly cancerous cells do it the most rapidly of all the cells that are there. So the period from cell formation to cell division, when it carries out its routine activities, and one of those, as well as whatever business it has to do, whether it's secreting compounds, whether it's making mucus, whether it's digesting products, all of those things that are going on, it carries on at the same time. It's making an extraordinarily large amount of preparations to divide. So it's the busiest period, and most cells spend between 75 to 90% of their life in what we call interphase. So we recognize it by effectively not seeing a lot of changes in the cell. You can find out about it metabolically, but it's not something you can visualize under a microscope or an electron microscope. Maybe the cell subtly increases or changes shape a little bit, but you don't see any really interesting things going on. And really what our focal point is the nucleus. But what's happening on a molecular basis is we really don't even see the DNA. It's in a state called chromatin, where effectively it is uncondensed. In other words, it's not tightened up or coiled. And so all you really are imagining is tiny little threads that you really can't make a whole lot out of. And so we look at the subphases, and this is where the controls, and this sort of dovetails down the road into why one of the things you're going to see is a delay. One of the things that radiation produces is this mitotic delay. And that's one of the things we'll consider next time. It's one of the first things. A little hint for reading a textbook. You have textbooks to study, whether it's an e-textbook or a hard copy textbook. I learned this technique a long time. It's called scan, search, and summarize. And the first thing you do is to get a feel for a chapter, you scan it. You look for things that are highlighted, things that are outlined, things that are separated, bold-faced, italicized, if they have any illustrations or graphs, and you kind of get a feel for them. Then you actually do the study, then you go back and try to do questions you formulate. It works. It's worked for years. I actually took a class in that over a summer when I was between 8th and 9th grade called How to Study. And it served me fairly well since I'm actually good at this stuff. So we're going to see fate of irradiated cells, division delay, then interphase death, reproductive failure. So taking a look at what's happening here. Uncondensed, interphase, and the subphases that are there. Should be very familiar with this, 3 phases. G1, known as gap, they call them gap phases, G or growth phase. Vigorous growth and metabolism. There are cells that eventually exit this and go to something called G sub 0 when they have reached their highest state of differentiation, where they're as organized and advanced as they could be. There aren't a lot of those. And these are the ones that are particularly unreactive to radiation or to cancer chemotherapy, to chemical therapy, or physical methods a la radiation, such as nerve cells and muscle cells. Really probably the best example of those. So we call that G, and we can induce with some of these things cells moving into this phase, not necessarily desirable. S phase, the phase that really is the significant feature for us and what it's doing. It's actually, this is when DNA is replicating. It's also when DNA is most easily damaged. We'll look at some of the damages there. And then finally, the second gap phase or second growth phase is a preparatory phase. And it's almost like it's almost the internal mechanism of the cell, reading the tea leaves or reading the signs to make sure it's ready to proceed to mitosis. And there's a number of ways to do it. And this is the beauty of this. And the first thing to realize, this is an area where we think of governance. Living systems are governed. They're slowed down. Inherently, they're quite rapid. So when we lose controls, effectively, this cell cycle would spin out of control. Your heart rate and my heart would normally be over 100. But if you are relatively healthy, your heart rate on rest sitting here now is between 60 and 80. That's because we have a system that governs that. The healthier you are, the better that's kept in check, and the more resistant it is to changes. So you can go do a vigorous workout, get your heart rate up, and come back and sit down. And if you're very healthy, that heart rate will come back to normal. That's a real good measure of overall fitness. It's there. So when we lose these controls of this cycle, and this is some of the events that you will see as we explore what's in that third chapter, what kind of goes on, because it alters the creation of certain chemical agents, mostly proteins, that impact the cell cycle. So what the cell cycle does, it doesn't proceed unless it goes past these effectively 2, but really there's 3. This is actually a double checkpoint. Not that that's terribly critical for us. Okay. So the first and the most important checkpoint, the so-called restriction point, as cells are growing and as they're getting ready, they're building up the building blocks for DNA, the building blocks for RNA, the building blocks for cell membrane, organelles, all the stuff that's in the cell. They produce a variety of products, typically proteins that collectively we call something called cyclins. And they bond with another protein, which is an enzyme called, as you'll see shortly, called cyclin-dependent kinases. When they reach a certain critical level, it'll release this checkpoint. It's almost like just pulling the switch so it'll allow the cycle to move to its next phase. That's the critical piece that allows cells. So if we lose those controls, they will just continue to accelerate and accelerate and accelerate. Ultimately, what is a cell that is damaged or a cell that doesn't work properly? It's a cell that has errors, typically errors in DNA. When do we make any kind of error? We do things rapidly. Think a zillion examples you could think of yourself, whether you're doing something at home, whether maybe if I'm running late to school, I'll forget to bring something. How many times have you forgotten something because you're doing something too fast? It's no different than any other analogy you can come up with. So that's the first idea of this restriction point. Then it goes through the DNA. And what really happens more at the second restriction point, this is a real governance about potential, because we make errors in DNA all the time. It's just they're not critical errors. If there's enough critical errors, then those chemicals are not going to be produced, and then this checkpoint won't be released, so it will not undergo. So it's almost like surveillance. Is the cell healthy enough to undergo mitosis? Are we likely to get a mutation? Is it likely to be a significant mutation? So again, more stuff like that. Little things. Okay. So the whole replication thing, I don't care about forks and bubbles. That's advanced and how that thing, but it does impact us with regard to, it kind of gives you an idea. If you look at illustrations of replication forks and replication bubbles, you can see the strands. And you begin to see where we can get damage to the strands, either single damage or double damage. That's very significant. Because isolated damage in DNA is relatively easy to repair, and it's easy to repair because of this stuff, DNA polymerase. In our cells, we have a very advanced form of DNA polymerase called DNA polymerase 3, Roman numeral 3. And the beauty of this, in our cells, it has the ability to not only detect errors in the assembly of DNA, and what are the errors? Again, it goes back to original DNA. The A's and the T's, the G's and the C's have to line up with each other. So if you have an adenine, an A, bonded to anything but a T, it's an error in DNA. Same thing, a G bonded to anything but a C, or vice versa, an error in DNA. So certain systems just repair that. They'll remove the error per se, but it's not very accurate. And if you have me for micro, we talked about it. It's called mismatch repair, meaning it's as likely as to remove an error as it is to remove the actual appropriate strand. It corrects, but it doesn't correct with a brain, so to speak. Whereas DNA polymerase 3 has a remarkable capability of identifying an error and removing specifically the error. Remember, it's a double chain. So is the error on the good side or the mutated side? Well, it identifies the mutated side, gets rid of it. So it's a good thing. And the directionality and things like that are noteworthy as well. And again, this is all based on the concept of semi-conservative replication, meaning whenever DNA is replicated, you have an original strand and a new strand. And what that does is it kind of keeps the material fresh. It's not like you're just copying and copying and copying, because copying over time degrades. It's like putting a piece of paper in a photocopier and then taking the copy and then photocopying the copy and then taking that copy, etc. Over time, it's going to degrade. So you may, and that's one of the, it's sort of the difference that was explained to me between semi-conservative replication, maintaining a lot of the refreshness or originality, versus cloning, which tends to deteriorate over time. And so here you are. This is that idea of bubbles. And the only thing that's relevant about this is that we begin to look at where the damage could be. So you could have damage, an isolated damage, one break. Again, comparatively easy to repair. One error in one of those bases, as compared to having a double error where DNA strands actually break. So a double break is much more likely to cause problems. It's a lot harder to repair. And indeed, it can cause definitive chromosome abnormalities, missing pieces. When you start missing pieces, then you begin to get these errors. And I'm going to review those very briefly because you should have had them on micro, but I'm going to review those with a little snip of another PowerPoint before I get to this other one that I found. Okay, and you know, all that type of stuff that's there. These are the fork is here, the bubbles are here. Who cares? You want to look at the animation, go ahead. So as you can see, most cells need to constantly replicate, other than ones that have exited the cycle. And you can see skeletal, cardiac, and nerve cells do not. When we damage those, and usually it's the surrounding tissue in cases of radiation or chemotherapy that indirectly cause damage to them. So the M, or mitotic phase of the cycle, it has to do with the actual separation, and you're probably familiar with all of those things that are there. So the different parts of mitosis you should be familiar with. I always use the little mnemonic PMAT, prophase, metaphase, anaphase, telophase, works for me. Use whatever you want. I'm all about these interesting little tools. We use them in medical school. Make no mistake, everybody uses these types of things to remember them, unless I'm somewhat gifted because I have a very good memory. And so certain events occur. So there's 4 major events that occur in prophase. The chromosome, the big one is we start to see the chromosomes. That's why everybody thought that's where the damage was. We're just being able to see. and the nuclear membrane disappears, the nucleolus, another organelle within that, begins to disappear. We start to see the spindle form. Those are the 4 events of prophase. And what happens is and when we get to the very end, what's called telo or telophase, those reappear. So there's 4 events of prophase that occur, and we reverse those 4 events in telophase. And they go crazy with identifying late prophase. The kinetic core is something that you see being a sort of protein bud attachment that eventually will allow these to attach to the spindle. Be something like over here and over here, these little kinetic cores or centromeres. And so, and you can go crazy looking at early, middle, and late, and I don't. Metaphase, they line up in the middle, big M for meta, big M for middle. Easiest phase to see. Anaphase, shortest phase, they separate. They're sort of pulled apart like that. And if you look at any slide, you can see those. Typically in the laboratory, we would use like fish egg slides and things like that. They were pretty easy to see. And then telophase, the reverse. And it's sometime around that point in time when telophase occurs. At the end of anaphase and the start of telophase, we begin to see the cell begin to demonstrate its own division, something called, as you will see, a cleavage furrow. And it's almost as though we're beginning, little underpinnings inside, which are called actin filaments, begin to pull and tug on the cell membrane and cause creases to form. And to me, the best analogy in the world are, I love this, balloon puppets. Love that. Mine always burst. Yeah. When you have children, you'll have birthday parties. There'll be balloon puppets. Trust me. If you have children already, too bad. And now you're stuck. Children are a pain in the neck. Grandchildren are fine. Fortunately, you have to have one to have the. You're stuck with that. Go figure. Why can't we go directly to grandchildren? And so these are the signals. The go and stop signals direct when a cell should and should not divide. Has to do with surface to volume ratio. That's really important. When cells are too large, what happens is there's not enough membrane to move stuff in and out. So a big room like this, if you were dead in the middle of the room, it would be very hard to move stuff from the ceiling to the floor or the walls to get to you. It's kind of like that. And this is something that we see lost, let's say in a cancer cell, contact inhibition. Cells, when they touch one another, it's almost as though there's a signal to stop. It's a physical signal to stop growing. And we see cells grow over and above. And these are the cyclins, the regulatory proteins that accumulate. Some of the radiation and the mitotic delays in mitosis directly related to this, because it's not making enough of those. If it recovers, then we see almost like an interestingly a rebound phenomenon when we start to look at the curves to see that. And the cyclin-dependent kinases, okay, these are additional chemical agents which happen to be enzymes, and they form a complex which basically releases sort of the stop sign, and allows it to proceed, and then they all disappear and we start all over again. So that's really the summary of this. And then again, I'm not going to go into all the DNA sequencing and things like that, but I said if you take a look at these, and we begin to see when isolated breaks, sometimes when things bond to these indirectly, sometimes when they bond together, which we'll take a look at. These are different things that cause irregularity in this biological zipper. And it's like having anything. You should turn that down. Is, like I said, from children. He's 37. He has to tell me he's getting blood work done today. That's a 37. It's okay. It's okay. Actually has a commercial driver's license. In order to maintain that, you have to periodically have health checks. He drives for. Nevertheless, you'll be able to see some of these things, and we will look at those. So I'm going to go. I will budge out of that and pick. And this is one I used for anybody who had me for micro saw this. I just want to go to a couple of things that, even though it deals mostly with bacteria and in different ways, but we take a look at these different kinds of mutations. There are, the language is really important about mutations. Let me get back to it. Mutations. We mutate all the time. They are referred to as spontaneous mutations. Whether it's a bacterial cell or one of our cells, if you are exposed to environmental factors, a la radiation, you will increase the likelihood of that occurring by a factor of at least 1,000. So when we're subject to excessive amounts of radiation for whatever reason, not just therapeutically, you're surely at the very least, and then we get into all the nuances about how high the dose is, how frequently, and again, that's where Dr. Buxton will come in. So occur randomly at infrequent characteristic rates, et cetera, but spontaneous mutations in our cells, because of the DNA polymerase III, are comparatively rare. Okay, and if they occur, they're relatively easily fixed, because most spontaneous mutations are really just errors, base substitutions. Okay, and so there's a variety of environments. So it's comparatively rare. So you can see an environment does not cause mutation, but selects cells that grow in it. Okay, and we see mutations are very significant when we deal with the microbial world, when we talk about bacterial resistance and things like that. This is a base substitution, or what we frequently refer to as a point mutation. One isolated amino acid is, you can see a G bonded to a T. That is, in some instances, that will not even be noted because it doesn't alter the DNA. It depends on where the mutation occurs. The universal language, we call the genetic code, typically, if the first two letters are determined what the amino acid is, if the third letter is an error, it generally doesn't have any impact. There's a lot of inherent safeguards that are within this. So it's an isolated change that occurs. So depending on the nature, and they give you a chart here that shows the 3 different kinds of mutations. Here's a typical base substitution. It can be silent, meaning that it does not alter the amino acid of the sequence. It can be a missense mutation, which means you incorporate a different amino acid and it alters the function. The classic example of that, of one amino acid, it can have a negligible effect or it can have a significant effect. And the classic example of a significant effect is hemoglobin S, sickle cell. That's a one amino acid error in the sequence that causes problems with that hemoglobin molecule, where it loses its shape when there's low oxygen. It can have significant impact whether someone has the disease or even just carries the trait, because they're going to have a certain amount of that. And we see that with athletes when they go to play in elevation, we have to be so careful. Guy played for the Steelers years ago, he can't shut up. He's a commentator on ESPN, Ryan Clark. He never met a microphone he didn't like. He loves them all. So he has to comment about something. But to be fair, the reason that he played there once and ended up rupturing his spleen and having to have a spleen in his gallbladder was the hemoglobin clotted and effectively developed significant blood clots. Life-threatening. Which never helped them when they had to play in Denver in a significant game. Nevertheless, he was like Troy Polamalu's running mate. So that's a missense mutation, and he just had the sickle cell trait, not the disease. Nonsense mutation is the worst kind, meaning that it will cause a stop code to be generated, and it will stop the protein from being assembled. And generally, the more of those you have, the very soon that becomes incompatible with living. As cells die and other things die. So these are the things we look at. Reactive oxygen species, we'll take a look at that are part of that as well. And there's a variety of things where you can induce these mutations. When we begin to see deletions or additions, this is the type of thing we see with radiation. Basically, we get things that either add or cut out, and what we call downstream, we tend to see a lot of big problems. Read that as stop codes. So I thought I'd mention that. Now, we've got about maybe 20 minutes or so left, and I wanted to show you this, and I'll send you the item. So it's over here. Oops, sorry. Much as I hate to take away from the kids things, I'll send you the where did I put it? Top. I'll send you this link that's there, the SlideShare link, and this is what I came up with here. So I'm not sure where it comes from, but it was to me it was fairly interesting. And so we start. These are just some of the things we're going to start to look at. These dose response curves. Maybe you had a look see with them with Dr. Buxton. Did you? You start to. He didn't do any of the curves yet. So linear, directly related to the dose. We're going to see some non-linear. This is talk about these shoulders, where you're going to see that depending on the dose and the frequency of the dose, sometimes you don't get a dramatic effect necessarily right away. Okay, threshold, the dose at which the effects are produced versus non-threshold. And so in other words, the threshold would be the degree of dosing. So there's a bit of curves to look at that we'll be looking at. I'm going to see if I can generate those with images that I can either copy and paste it to like a PowerPoint or just like a handout type of thing that I can do. So there's a little bit of work that has to do. And this a term I was not familiar with, the stochastic effects occur by chance, usually without a threshold level, increase with increasing doses. Again, a lot of it's dose related. The severity of response is not proportional. Okay, so mutations in cancer, the two major. Deterministic effect, health effects that increase in severity with increasing doses above threshold, high doses delivered short period of time. And these are the kinds of things that, why the guidelines are present for dosing and things like that. Erythema is relatively common. That's just redness. You see a lot of that. I mean, all familiar with erythema, every time you get sunburn, that's just the base sunburn. That's just an increased circulation of the skin. And if it gets more significant, the degree of irritation or inflammation, and you might get separation, blistering, second degree burn, etc. Okay, cataract formation, obviously a big player. When you administer some of these things, do they use eye protection? I've never had it done. Anybody know? Do patients get eye protection when they do this? I'm curious. I don't know. Hey, I'm happy I've never had it done. That didn't help. Why can't I see this? I must have done something dumb. Well, I can't read that at all. Can you? Hold on again. I'll see if I can do a reload on that. Yeah, here we are. So you can see that all right? It must be, so you can make that out fairly easily. Radiation effects, cellular level, changes in critical or target. So you're looking at the DNA strand. It's about the DNA. That's there. This is where you're looking at it. RNA is remarkably resistant to problems. We looked at this, the idea of direct and indirect effects. Direct effects are the DNA exactly about the radiation. Okay? So, as you can see, because of its mass, is more apt to cause damage to DNA by this direct effect. Other molecules contribute, such as RNA, proteins, enzymes, may be affected, but less likely. Okay? So, most of the damage to DNA molecules from X-ray is accomplished through the indirect effect. That's the effect it has on water. And since, whatever, depending who you read, 60 to 75% water, we're a great big old bag of water. There's a lot of damage to the water molecules that are there. Okay, so water molecules ionize, they develop free radicals, which in turn, those free radicals indirectly do the damage, although it's very significant. So you form these toxic forms of this, and then we'll get into the oxygen later. We're oxygenated tissues because of that. The effect on oxygen is significant as well, where we get toxic derivatives of oxygen. So, there's what to be talked about with regard to that. Here's the free radical that has an unpaired electron. That's the definition. If you remember that from basic chemistry, and its valence shell the outermost shell, or whatever you want to orbit, of electrons. These free radicals aggressively join with DNA to produce damage. In the presence of oxygen, the hydroperoxyl free radical is formed, and that one is particularly nasty. So that's when we look at the degree of oxygenation of tissues as being impactful, something we get to later in the context of these presentations. Okay. So there's the direct versus the indirect effects that are there. And so the indirect effect mostly will start with the radiolysis, separation of water. As a result, the living body consists 79%, again, depends on the author. Ionization of pure water leads to fast-moving free electrons and positive charge molecules. And these radicals cause significant damage. So the big deal, can you see that all right? Because again, I don't see it quite as well from where I'm. Obviously, you'll see it on if you review this presentation, and you should. Changes or loss of bases, that base substitution. Again, the ability for DNA to open and close is based on what if it was single and isolated, a very insignificant bond called a hydrogen bond. But the more you have of those, okay, the greater the effect is, because that's what allows that molecule to open and close. Really, that's what allows it to replicate. So disruption of the hydrogen bonds, very significant. And then the breakage, and then occasionally we'll see cross-linkage of DNA strands within the helix or outside of it. So you can get, it's almost like getting jammed up in the middle or attaching to things on the outside, if you want to think of it that way. So I'm just thinking about all the things, you have a favorite, jacket or something, and the zipper kind of dies. Something gets stuck, hate that. If you're starting to pull it down, you got to take it out over the top of your head if you can. When I used to weigh about 280, that was very hard. There's a lot of obstruction. Don't laugh. It's in the FBO, fat, bald, and old. Same jokes, I've been using them for 1,000 years. So they can be undamaged. And now and this is another topic, whether there's, and this is part of this third chapter, whether it's lethal or sublethal injuries. Cells are damaged by the ionization, but the damage is repaired. Mutation, alterations in DNA, incorrectly repaired, the cell function is altered, etc., or leading to a cancer cell death. Cell damage is so extensive, no longer able to reproduce. And very, very often that's our goal, but it can be a side effect as well. Here's the sublethal. Damage to DNA. Typically, the sublethal ones are the single strand breaks. Again, they're easier to repair. The enzymes, read that as the DNA polymerase 3, recognize the damage and coordinate the removal of the damaged section. That's the beauty of it. You can target the section that's damaged, not the other section that's healthy. Okay, and then replaced. The cell cycle, they reiterate that. So it's a nice little, for whatever reason, I happened upon this morning. Just looking at, I think I Googled radiation biology PowerPoint, and this popped up, and I went through it. So when the cell is irradiated during G1 or early S, the damage will be duplicated during DNA synthesis and result in breaks in both arms of the chromosome at the next mitosis. So timing becomes an issue as well. And we'll understand a certain number of cells are in that particular area as well. Cells that are more easily damaged, radiosensitive, this is the idea. Are they very, very rapid reproducers? All those lining cells, and I'll do a section on tissues where I'll review the different lining cells and explains why the problems that your patients are going to have, because they're going to ask you what's going to happen to me afterwards. Am I going to lose my voice? Am I going to have diarrhea? Am I going to have irritation in my skin? Take it from somebody being around for a while. Nobody likes surprises, other than for your birthday. Other than that, nobody, when it comes to healthcare, you don't want surprises. Found that early on with patients. So I can remember it's, someone saying, asking me a question about, is this going to hurt after the surgery? Yes. How long? Okay, you tell them. Overestimate. Oh, it's going to be a pain in the neck for 6 to 8 weeks. 2 to 4 weeks, they're better. They go, oh, it's wonderful. You're a hero. Rather than telling them you'll be back to work in 2 weeks, and then 8 weeks later they're not. They go, what did you do? We like to refer to as the WTF factor. Just saying. So how rapidly reproducing, the degree of differentiation if they're immature or not terribly advanced, metabolism, how busy that cell is. The more oxygen it utilizes, the more water that's going to be in the area. All of those add to the picture. I found them in my readings. The most interesting one was, now erythroblasts are a kind of a stem cell. They rapidly, germ cells are like that. Base cells of skin and lining, very highly. Lymphocytes are a mature cell. And Dr. Buxton and I were discussing it yesterday. We were sort of reviewing where we're heading with some of the stuff. And a lymphocyte is a form cell, but it has a very, very limited lifespan. And I think that's why that a mature lymphocyte is so sensitive. It doesn't last very long. Like if you produce them for an infection, they have a relatively limited lifespan. Not all of them, some of them. So radio-resistant cells, not susceptible to damage. Okay, low reproductive rate, well differentiated, low metabolic rate, the opposite. Nerve and muscle cells basically are out of it. And now this looks at the different intracellular structures, the nucleus you're familiar with. So what happens? So in the nucleus, fine, cell division. Cytoplasm, increased permeability of potassium and sodium, active transport. Nucleus is more sensitive than the cytoplasm. Obviously the DNA. And then we worry about chromosome aberrations. So rapidly dividing cells, such as when you talk about hematopoietic, making red cells and white cells. Of course, and this is why we see bone marrow, which is where this is located, often being affected significantly. Okay. So you can see size of the irradiated tissue result in mitotic delays, which we'll talk about later, and cell death. Okay, and then they look at the different mechanisms that cause it. DNA damage, mutations, and/or typically chromosomal damage would be truly DNA damage. Breaks in the chromosome. This is interesting. Bystander effects. Irradiated cells that are damaged release chemicals because the cell is damaged, stuff pours out of it. The bystander effect occurs upon irradiation by particles or X-rays. Okay, and what effectively happens, the cells that are in the neighborhood get bombarded by chemicals. So they're talking about what ordinarily you might think of as an innocent bystander being affected by something like this. And then apoptosis, programmed cell death without necrosis. Cells round up, draw away, and condense. So the bystander effects are interesting as well. And cells recovery from the damage and bystander effects involves a variety of repair mechanisms, etc., and related to the dose. So we look at different organs, and you're able to determine which organs, and we'll look at this as well. That's part of it. That's one of the things. Short-term effects we see early on. And so again, when they talk about parenchymal cells, it means a specific tissue. Like, do we see it in the kidney? Do we see it in the liver, etc., specific tissues? Okay. So bone marrow and mucous membranes quickly, rather than muscles, which you hardly see any at all. And then the long-term effects that are there, and what happens to some of these, we begin to see the gradual replacement in certain tissues of scar tissue, and scar tissue is no function. So death of replicating cells, damage to the fine blood supply, which plays a pivotal role. So that's also part of it. And then some things that modify it, and this, again, type of things that you look at more when we look at the dosing, the total dose, the the timing of the doses, the dose rate. And then this is an interesting one that, again, we'll look at with certain kinds of tissues later in the term, more pronounced in the presence of oxygen. Because now that hydroperoxyl, what we call the superoxide free radical, very, very nasty free radical. That's there. And then the way the energy is transferred and the different particles, and again, that's outside of my paper, so to speak. So treating radiosensitive oral malignant tumors depend on factors, all the different things that are there, and that's just part of it, and how you sort of fractionate or divide the radiation. And then they look at some of these, and so you get some ideas of what the effects are. So, and I'm curious, and I wanted to mention this one just for instance, that it doesn't always have to be directly from radiation. So, redness and inflammation of the lining, formation of what they call a pseudomembrane, difficulty in swallowing or eating. This is, I'm actually taking medication for this. I had a reaction because I was taking it's not an anti-cancer drug per se. It's a biological that suppresses the inflammatory response because I have an inflammatory arthritis. You see it advertised all the time called Cosentix on TV all the time. So I was taking injections of that once a month. All of a sudden, I had a tongue. I couldn't hardly swallow anything. It was like a terrible sore throat. Nothing tasted right. I actually had a yeast infection because normally your inflammatory response clobbers yeast, which most of us have in our mouth. So it only flourishes with somebody if they're on broad spectrum antibiotics or they're taking anti-inflammatory or having those kind of injections. And so it produced that kind of problem associated with it. So it doesn't always have to be radiation, but that's another example. If it destroys normal cells or normal mechanisms, you have abnormal phenomena. And so, and the same thing, problems with taste buds, which are fairly sensitive, but they do return for different parts of the tongue because they have different sensors. In anatomy and physiology, we explore whether it's the anterior third, the middle, or the posterior third of the tongue, and see how long the recovery rate is. They'll repair, but you're looking at a 2 to 4 month recovery period. And the same true here for salivary glands. I'll stop here. I do want to finish these last handful of slides down the road, and then I'll look more into the other when we get together back in on Thursday. So thank you.